Hi, everyone, and welcome to podcast 14. We are going to take a break from all things molecular in nature and talk about things on a larger scale, and we're going to talk about animals. The reason for that is we are going to periodically visit animals over the course of our various units. We're going to come back each time and talk about relevant animal organ systems. So we will be talking about them as we go through things throughout the year. But in order to do that, we probably need to get a little perspective on exactly what it means to be an animal. And so that said, let's remember that the animal kingdom, like all kingdoms, is just a level of classification, a taxonomic level of classification, and that is a human construct. It is an artificial definition. The characteristics that we look for to qualify something as an animal are precise and compartmentalized, and Mother Nature isn't like that, really. Mother Nature flows along a continuum from the most simplistic bacterium all the way to the most complex animal. And we file all these different organisms into little compartments because it makes it easier for us to deal with them. But that is not how they are in reality. It helps us make sense of them. It's useful, but there are all kinds of shades of gray in between these discrete compartments we create. So that's why taxonomists, scientists that deal in classifying organisms, have a lot of stuff to figure out. It's not always easy to figure out where an animal or plant or whatever goes in one of these little discrete categories. So just keep that in mind, and we will flow from there. Okay, so what is an animal? Well, essentially, an animal is an organism made up entirely of eukaryotic cells, cells with nuclei. Their cells have no cell walls. We're not going to worry about that statement right there, but we are going to worry about that all animals are heterotrophic, meaning they must consume food to get energy, unlike autotrophs who can make their own. They are ingesters, meaning they take food in and then digest it inside their bodies. There are heterotrophs that do it exactly the opposite. Fungi, kingdom fungi, for example, is a digester-ingester. They break stuff down outside their bodies by secreting digestive enzymes on it and then slurp it up. So there's two ways to do it. We do it the ingest-digest route. All animals reproduce sexually. This does not mean that animals cannot reproduce asexually. Some animals can, but all animals can reproduce sexually. Okay? So in their sexual reproduction, or in general, the diploid stage of an animal is the dominant stage of life, the stage that you see commonly out when you are looking at an animal. Now, what diploid means simply is that you have one set of chromosomes, one set of genetic material coming from the father, and one set of genetic material coming from the mother. That's one set plus one set equals two sets in the baby, and that's diploid. And so that baby will grow up and have two sets of chromosomes of genetic material from its mother, and one from its mother, one from its father. It is a diploid organism. And so that is what you are, a diploid organism. The only things that are not diploid in us are our sex cells, our gametes. We cut that chromosome number in half, and we'll talk about that much more when we get to genetics. But the diploid stage is the common state of existence for us, except for our sex cells. Development. Now, this is where it starts to get critical. These are some concepts we need to understand how animals are classified and why they're grouped the way they are grouped. When this haploid or single set of chromosomes in the sperm meets this other single set of chromosomes in an egg, we get a fertilized egg, or a zygote, that single cell that is the beginning of a new individual. Okay? Now, that zygote is not going to stay one single cell forever. It is going to begin to divide. And that division that that cell undergoes is referred to as cleavage. It is mitosis, and we will talk about that word much more later on too, but you should have heard it before. Mitosis is simply cell division, dividing the cell that results in two cells that are essentially identical. Okay, so in this picture, this zygote, I lost my pen, what a surprise. My zygote has divided once, then twice, and I'm trying maybe another time, but we have gotten 8 cells, it looks like, out of this division, okay, and 4 on the other side. So that is cleavage. It's just a division of the zygote over and over again. Now, that division goes on. We don't just stop at 8 cells. The 8 cells divide, and then 16 cells divide, and so on until we essentially get a ball of cells. This ball of cells that may be, maybe 100, maybe a little more. It depends on, maybe many more, depending on the organism. This is called a blastula. Blastula ball of cells. Okay? Now, something important about this ball of cells, it is hollow. It is not a solid ball. It's a hollow ball with a space inside. So the zygote here divides until we get 8 cells, and we get a bunch more cells after many divisions, and we get this blastula, this hollow ball of cells. And you can see the space inside called a blastocele. You don't need to know that, but they've cut the blastula in half so you can see that it's hollow inside. Okay? Now, in this blastula, obviously, it's not colorful like this. It's just a ball of cells. But scientists have determined that there are cells in certain positions in the blastula whose fate is kind of determined as to what type of tissue they will eventually become. Cells in this position in the blastula are going to become what are called ectoderm cells, or cells that lead to things like your epidermis, your skin, and stuff like that. These cells here in the red have a different fate. These cells in the yellow have a different fate. These cells here have another fate. Usually we don't separate these out from these ones in the blue up here, but this diagram happens to. So, but understand that these cells at this point are getting committed to become certain types of tissue. Up until now, they're pretty general, and if you plucked a single cell out of that ball of cells, it could pretty much become another embryo again. We'll talk more about that later. But right now, these cells are beginning to commit to grow up to be certain types of tissues. Okay? So, how do we get there? How do we get these things to becoming types of tissues? Well, the blastula, this hollow ball, is essentially going to turn itself inside out. And it's going to make a structure called not a blastula, but a gastrula. Okay? That would be gastrula with a G. So blastula with a B comes first, gastrula with a G comes second. Now, the process in which this blastula hollow ball turns itself inside out is known as gastrulation. So the inside out blastula to gastrula transformation is called gastrulation. Okay? Now, this is the actual process that is going to give us our initial layers of actual tissue. Up to now, we're just seeing a bunch of cells that have no tissue organization. After this thing turns itself inside out, we are going to see the first embryonic tissues emerge, and those are going to be what lead to all the adult tissues and adult body parts. So, here we are, our zygote, cleavage. Cleavage goes to blastula. Blastula here turns itself inside out, and that's what these arrows are indicating. These cells are going to begin to move towards the inside like that, and we're going to see this thing called a gastrula be created. Okay? Now, the hollowness of the blastula is not trivial. It's not just, oh, big deal. It is the space that allows this inside-out movement to occur. If the blastocele, if that hollow cavity wasn't there, the thing couldn't turn itself inside out. Now, here is the blastula here with its hollow space. Okay, now what happens is, right in here, it starts to come inward and into this hollow space, and in fact, the hollow space begins to collapse. And if you look here, see how big the hollow space is there? The hollow space has gotten much smaller here. But this hollow space has gotten much bigger. This hollow space here is replacing that hollow space that's getting squeezed out. This hollow space is called an archenteron. Archenteron. And that is really important because that is going to become the organism's gut, its digestive cavity. This is the inside of your digestive tract. This was you. Believe me, you were a zygote, you went through cleavage, you became a blastula, you turned yourself inside out, and that's your gut cavity, where you digest your food, called your archenteron. Okay, so archenteron is the future gut cavity, and it forms during this gastrulation process. Okay? Now, after gastrulation, then we get more development, more refined development of the tissues, and you can start to see tissues and actual almost recognizable body structures form. And eventually, some animals actually develop directly into adults, but some go through what is called a larval stage. And a larval stage, like this one, and everybody knows this is a tadpole larva, I'm sure, is when there is a immature form that is really different from the mature form. Think caterpillar to butterfly. The caterpillar is the larva. It is just fundamentally structurally different than the adult. You are not fundamentally structurally different when you're a child from your parent. You just look smaller. But this thing is going to undergo really dramatic changes to become the adult. It's going to lose the tail. It's going to grow feet, and so on. So that's the distinction. They eat different food. They live in a different habitat. They're quite different. Until metamorphosis occurs, and then we see the adult form that we would recognize. Okay? Not all animals go through that larval stage, of course, but many do. All right. Now let's talk about a history of animals, animal evolution. Now, we do not see complex animals until very recently in the history of Earth. If this is a time scale, with this being the Earth is formed, and this being right now, this is now, okay? We don't see any evidence in the fossils of life till maybe, I don't know, let's say here. We don't see any eukaryotes at all, and I mean just single-celled ones, let's say, I don't know, maybe here. And we don't see any multicellularity to create animals until, golly, probably, I don't know, somewhere up in here. So multicellularity is quite recent, geologically, evolutionarily speaking. Okay. All right. Now, who might have been the common ancestor of the animal. Now, again, this says diversification began more than 1 billion years ago. We're talking going back to very, very primitive single cell or very slightly multicellular creatures. This is a modern day organism, and it is called a choanoflagellate. Okay? It is the protist. Kingdom Protista is kind of the junk drawer of the animal of the world of life, the eukaryotic world. If scientists couldn't identify an organism as a plant or an animal or a fungus, they would toss it into the Protista drawer. And so the choanoflagellates are a type of protist, but they have the most primitive animal-like characteristics. They are not animals. They are protists. They don't possess all the animal characteristics, but it is thought that the common animal ancestor to all animals that have ever arisen probably started out looking something like this modern day protist. Okay? It was colonial, meaning it was made up of individual cells that just kind of had an agreement, if you will, to live together. They weren't multicellular truly. Much like certain bacterial colonies today live together, but they're not multicellular, if you will. So, one hypothesis is that animals originated from some flagellated, meaning a protist that had a flagellum, okay, or many, as the case may be, a colonial protist that had flagella, and that it kind of made over evolutionary time these kinds of steps, and eventually, due to some folding that occurred, to the creation of this hollow cavity here, took on the animal-like characteristics that we associate with animals today. No one was there, no one saw it, but this is one hypothesis about how this might have occurred. Okay? Now, this is what we need to know to really talk about animals as we need to for the purpose of the course and the purpose of understanding their organ system. We need to understand their body plans. Okay? Features of body plans that we need to be aware of are symmetry, how many tissue layers are present, what type of body cavity, if any, is present, and the type of development that the animal goes through. Okay? And when we understand these basics about just basic animal body plans, then it allows us to kind of dissect them in terms of layers of complexity, levels of complexity. So let's talk about symmetry. That is an animal, believe it or not. I'm sure you recognize that as a sponge, and if not, there you go, meet sponge. And this animal has no symmetry. It cannot be divided in half and produce two equal halves. It is totally a unequal, unsymmetrical blob. And if you cut this in half, these two halves are not going to be the same. That's the most simple type of symmetry, not having any. Okay? Sponges are extraordinarily simple animals. They barely meet the criteria to be an animal. They are only multicellular in the loosest sense of the word. Now, whoops, going wrong way. All right. Radial symmetry is the next type. You need to think of it as like a flower pot. It's a really helpful way to think of it. This is illustrating a little critter called a hydra, but you can see in radial symmetry, you can cut like a pie the animal in multiple directions and get two mirror image halves. I can cut it in this plane and get two mirror image halves, this plane and get two mirror image halves. I cannot cut it this way, though, and get two mirror image halves. Okay? So there are two distinct surfaces, the top and the bottom are the oral, the mouth surface, oral, and the aboral, meaning away from the mouth surface. Okay, there's no left and right. There's no head and tail. All right, think sea anemone, like in that, or a jellyfish, a moon jellyfish, like we commonly see down in the Chesapeake Bay. So those are animals that demonstrate radial symmetry. These are pretty simple animals, and it's pretty simple animals, generally speaking, that are going to demonstrate this type of symmetry. The last type is bilateral symmetry, and that is symmetry in which there is just one plane, that will give you exactly two mirror image halves. With this type of symmetry, you get body regions that we would call dorsal and ventral, or if this is a, let me draw a fish. That's easier to talk about, I think. Okay, if I cut the fish down the middle, now that doesn't necessarily give me mirror image halves. In this case it does, but the bottom, the belly, is the ventral, the top is the dorsal. Okay, I can get left and right, as you can easily see over here with the butterfly, left and right halves, and I can also get, if I cut it another way, an anterior or head end and a posterior or tail end, and that we only get those kinds of divisions in bilateral symmetry. Additionally, with bilateral symmetry, we see the sensory equipment, the sensory organs, generally concentrated at a head end of that central nervous system. Our eyes, our nose, our hearing, and all of that. This is a word you should remember. It is called cephalization, when you concentrate all those sensory organs and a brain at one end of the organism. It is thought to be a more efficient way to encounter the world, and a fairly significant evolutionary step to have those sense organs being the first thing that encounter your environment. You're not just haphazardly kind of like a jellyfish out there anymore. You have a sense of direction and a way to face your environment. So this is a summary of all the symmetry types here. You can see here the definite anterior, posterior, dorsal, ventral, left, right, and all that, and just the lack of those kinds of things up here with the flower pot, sea anemone type arrangement. All right, now, tissue organization. First, we need to know what tissues are. Tissues are simply groups of cells, but the cells are similar not only in how they look, but in what they do. Okay, so they do something that is similar. Your blood is a tissue, and the cells in there are similar. They're all doing a similar job. Muscle tissue is a tissue. Similar cells doing a similar job. Okay, now, some animals are so primitive that they lack tissues. Guess who? Sponges. Okay? They are really essentially aggregates of cells, of individualized cells living together in this unit. They function nearly independently. They're not completely independent, but they are nearly independent in their function, and they do not have this unification of a tissue type construction to them. Now, in animals that possess true tissues, which is pretty much everything except sponges, we begin to see a layering of tissues, and we begin to see it happening at the formation of the gastrula, and we call the first embryonic tissue layers that we see germ layers. Germ just means like the seed, the beginning. So these are the embryonic tissue layers, or the germ tissue layers. Okay, and these embryonic tissue layers will form the various tissues and organs of the adult eventually. Okay, and we saw them in that diagram. There are three, count them, three embryonic germ layers or tissue layers. They are ectoderm, endoderm, and mesoderm. Ecto means outside, so skin type coverings. Endoderm means inside. This is going to be lining the gut cavity, and mesoderm means middle. So that's in the middle of all of them, and we'll talk more about that. But ecto, endo, and mesoderm. Okay? Now, animals can either have 2 tissue layers or 3 tissue layers. If you are diploblastic, guess you have 2. 2 germ layers. That's it. So you're still pretty simple because you've only got 2. Ectoderm and endoderm are the first 2 layers to show up. The only animals that are really in this group are the jellyfish and the sea anemones and the corals. We'll talk more about them later. Triploblastic animals, however, have all 3 embryonic tissue layers to show up. And pretty much everything that's higher than a jellyfish would have this. So with the you and me and everything that's not a jellyfish is going to add the mesoderm to that ecto and endoderm and be called triploblastic. Okay? Now, the ectoderm covers the surface of the embryo and also, and this is not trivial, gives rise to the outer coverings, the skin, the epidermis, the central nervous system, which is what CNS stands for, the central nervous system. So the ectoderm is skin and the nervous system, kind of wildly different things, but nonetheless. The endoderm is the innermost germ layer, the innermost tissue layer. It is going to give rise eventually to the lining, the coverings on the inside of the gut, like the inside of your stomach, but also organs like your liver and your lungs. Again, kind of wildly different organs, really different functions, but this endoderm gives rise to things like that. The mesoderm is a layer that is sandwiched in between the ecto and endo. It makes sense. Meso means middle. And this layer gives rise to things like muscles and essentially most of the organs that are somewhere in between. If this is your body, here you are. I know you think you're better looking than this, but okay. And here is your gut. But you think your gut's better looking than that too. So anything that is between the gut and the outside, and this is an oversimplification, but anything that is in this space is probably formed from mesoderm, like muscles, bones, things like that. Okay, so this chart is kind of a nice one. It kind of lines all this for you. There is the zygote to the blastula to the gastrula, and the gastrula, when that happens, we get ectoderm, endoderm, mesoderm, and this is telling you what comes from each of them. Ectoderm gives rise to epidermis, brain, nervous system. Here we go with the endoderm, endoderm, digestive tract lining, respiratory tract, glands, which liver, pancreas, the mesoderm. You don't know what this is yet. We will. Somites are the beginnings of muscles, and here's some other stuff. Hopefully no one would ever ask you to go into this much detail. Bones, cartilage, we kind of mentioned that earlier. I would kind of know these. Circulatory system. So if you can kind of know these two here, that one, maybe this one, maybe lungs, and then muscle and bones and circulatory system, I would surely think you would be covered. Anyway, that kind of is a summary diagram. All right, I'm going to stop it here because this is going to get incredibly long. I can see it coming, so I'll stop here. take a break, and I will catch you when we finish this and talk about body cavities and some other important animal features. Go rest your brain. I'll see you back here in a little bit. Bye.